See the person talking. Did you eat? Did you eat? Yeah, to kick you.
Balance this out.
Get out here. And it's because of the cake I didn't go. It's because of the cake. Even as I'm sitting like this. Chicken take one. Have anyone come on, Adam and Eve, come on, Eve and the Satan. And I'm going to take the phone to call her. I'm not going to say that when I told myself tomorrow, Zanji, just hold it for today. Tomorrow you go.
Where is that?
In a meeting. He's been on it for a while. Maybe God is just trying to save me. But you know, even she that here she was saying that I'll bring a cake for you. Which one do I bring? She out here. She know just one. I said, okay, the usual. I pray that she yields to temptation and bring more than the usual for me. Because of that cake, I don't eat again for the rest of the day. After that lunch cake, I can't eat anything again. Because you're very particular about what your body. But right. You know, it's so my sister did it and brought it maybe my birthday or my husband's birthday. I just stay away from it.
Have you left the place?
Then I get out here. My God. Let me do and leave this place. But on Saturday that we're leaving. But it's like sleep mama will not be going back.
No, she actually drive on people, but she is spending up on Saturday. Oh, you can stay in her house. There's no, you're going back on Saturday. So he's coming back on Saturday. Okay. Very good. She actually done about now, but it can stay one. You get one advantage if you have motor office and got comfort as well. But that's all these police and couldn't know about. Yes. Hello? Yeah.
Eastern Nigerians, you have a meeting online or class online. People wait till, is it on the time before they join? I'm used to joining 15 minutes, 10 minutes before the time. Because anything can happen. No lectures. This your UG students, postgraduate.
I give them an assignment. So you I have AI check architecture. Yeah, some of them came on 93% and 5%.
That means because you could unpanel the brain as well. When eating AI detect when you can add Oh, Quillbot. Okay. It's free, but
You know, but to get that aspect, so you have to subscribe. It's around 15.
okay. These students are wonderful. Nobody's telling them don't use AI, but use your brain.
Yeah,
Use your brain.
You
Yeah.
can just use the
Or support. Eh,
People are not interested. So they
Kai.
do. No, generation now is also generation now is very big. You don't say anything.
Gen Z. Are you part of them?
Yeah, they know, they are about the good of it. No, you notice you start teaching these students. You know, students in Nigeria can do a class of stuff. You teach them, I don't teach them, so they can't spend on mic and every time like because you're just looking for where to even assist the student to do something better.
Hmm. Okay, at least one person has joined. What's wrong? There's hope. But the good thing is that they really attend, which is quite impressive. Hali na uni. She's the first to join. Itakuladi. Sala. Matthew. Jennifer tries to. They're good students. I mean, it's been a nice class so far.
Hi, everyone.
Hey, Dr. Chinwe, good afternoon.
Good afternoon, Dr. Grace.
Good afternoon, Dr.
How are we doing today?
Yeah, so we're good. The students are yet, I think we only have one student that has linked up. So you please bear with them. They will soon join. Okay, two. The class rep has joined. Wow, nice to see you. Welcome back. Great. Welcome back.
Thank you. Thank you so
Yeah. Sorry, we'll just give
Okay,
a few
so one
minutes.
of the things you have to tell your students is that I
Mhm.
don't do African time because
You
I don't
don't?
like to eat for people. I think it's a waste of my time.
Exactly, exactly. So class rep, you're hearing, ba? Hmm? Laddie. Hello, Laddie. Can you hear me? Maybe my volume is too low. Hello, Laddie, can you hear me? Laddie.
I can hear you, so I don't know if he can hear or he
I
can't
can
hear.
hear you, ma. Good afternoon, ma.
Okay.
Yes. Saba, so you've heard our
Yes,
guest lecturer saying that she doesn't like being late, people being late to class, which I've been
yes,
telling you guys, right? It's the vet training doesn't permit lateness, really.
that's correct, ma. I just sent a message to our common group chats, so more people will be
Okay.
joining in the next 2 minutes.
So I'm not going to wait for them. I'm going to start the class because I have
I think,
other things to do.
yes, and it's being recorded, so I mean,
Yeah.
yeah. Thank
Okay.
you so much for, yeah.
Thank you again, Dr. Grace, for inviting me to your class. And
Thank you.
hello, everyone, all the students. Maybe Dr. Grace, you can do a little bit of an introduction, and then I will
Yeah.
want the students to say their names so that we can start.
Okay. Let me just, I hope you guys can see me. In a hall in Abuja. I can't see myself. Can you guys see me?
Yeah, we can see you either. It's a blank screen.
Oh, wow. Okay. All right. So bear with me. So
That's fine. We
so we
understand all
have the pleasure we have the pleasure of having Dr. Chinwe Chukwudi, a very, very famous molecular biologist, but primarily a veterinarian. Her first degree is in veterinary medicine, University of Nsukka and Nigeria. And then she went ahead to do her PhD in the Royal Veterinary College, London. And she actually has her own lab that I think she would talk more on that, but she will be the best to talk on that. But she's got this vaccine lab that she has just established in the University of Nsukka. So she's actually making us very proud, you know, being that she is a vet, and then she's really launching into the molecular biology and not just stopping at that, being a master in the field as well. So you guys are really privileged to have her. She just got back from the US because she is she has an NIH active grant. She's been on it for some years, and so you have a lot to learn from her. I have a lot to learn from her. So it's really a privilege that we have her to take us on this next topic of nucleic acid separation, labeling, etc. So thank you very much, Dr. Chinwe, once again for accepting to teach. And ever since I met you, I've been challenged. And I believe that my students will be challenged as you take us through these topics that we have given you to help us with. Thank you very much. So,
Great.
we're 6 now. Yeah.
Grace.
So you can start, Ma.
Right.
Thank you very much.
Okay. I'm just trying to get to the particular slide that I want to be sharing. slide. Hello, everyone. Can you hear me? Have I gotten lost?
We can hear you, ma.
Can hear
Okay.
you.
Can you see my screen?
Yes, we can see your screen, ma.
What can you see?
Separation of DNA molecules, labeling, and detection of DNA fragments.
Okay, great. I just want to be sure I'm sharing the right screen because I
Okay.
have so many things open. And good afternoon, everybody. Once again, I would want all of you to introduce yourselves. Just say your name, how it is pronounced, and just one or two sentences about your background, just so I get to know who I'm talking
Sorry, Electrophoresis.
today. All right, just go. Anybody that wants to talk very quickly. Let's do this.
Good afternoon, ma. I am Watrahel Samson Shelia. I'm a vet, first degree vet. I did my master's in public health, veterinary public health and preventive medicine. I'm currently doing my PhD. It's nice to meet you.
Great. Sorry, what? I only heard when you said Samson. Which one is your first name?
Watrahel.
Watrahel. That's your first
Yes.
name.
Yes, ma'am.
All right. Next person. Anybody?
Good afternoon, ma.
Good afternoon.
I'm Jennifer Namdi Ibe. I'm a vet, first degree at Bello University, and I'm currently running my master's degree in University of Jos, Public Health and Preventive Medicine. Nice to meet you, ma'am.
Nice to meet you, Jennifer.
Yeah.
Yes,
Good
next
evening, ma.
person. Good evening.
Good evening, ma.
Good evening.
I am Naason Boyi. I have a first degree in veterinary medicine, University of Maiduguri, and I'm doing my MSc in One
Sorry,
Health,
which one is
University
your
of
first
Jos.
name? Which one is your first
Naason.
name? Nasan.
My first name is Naason, yes, ma.
Nasan. Okay.
Boyi
All right.
is the second name.
Okay.
Yes, I'm
So
doing my
for the
MSc.
for the rest of you, just say your first name. That's all I need to know so that we keep it simple.
Okay.
Okay. All right, go ahead, Nasan.
Yeah, as Naason is the name, my first degree in veterinary medicine, University of Maiduguri, and I'm doing an MSc course in One Health, Department of Public Health and Preventive Medicine, University of Jos.
Okay. Nice to meet you, Nasan. Next person.
Good afternoon, ma.
Yes, good afternoon.
I'm Ladi Amos Chabri. My first degree is in veterinary medicine, University of Maiduguri. I did my MSc in veterinary pathology, Ahmadu Bello University, Zaria. And now I'm doing my PhD in public health, University of Jos.
Okay. Nice to meet you, Ladi. Next person.
Good afternoon, ma.
Good afternoon.
Good afternoon, ma.
afternoon.
Yeah, my name is Idoga.
Sorry, your name is what?
My name is
Idoga.
Idoga. Yes, ma. I did my first degree in biochemistry from the Federal University of Agriculture, and I'm doing masters in One Health.
Federal University of Agriculture, where?
Makodi.
Okay.
No,
That's Futmina. No, not
no.
Futmina. Federal University of Agriculture, Makadi. Okay.
Just-
Okay. Okay. Right. Next person.
Good afternoon, ma.
Good afternoon.
My name is Matthew. My first degree was in veterinary medicine from Ahmadu Bello University, currently running One Health in University of Jos. Thank you, ma.
Okay. Welcome. Yes, next person.
Good afternoon. My name Abel, DVM AB Uzaria, MSc on Health. Thank you.
Right. Is the MSc in view or already done?
In view, ma.
Abel. Okay. So you don't have it yet. Okay. All right. Next person. Are we all done?
I think that will be all for the numbers of participants we have online, ma.
Okay, great. Dr. Grace, I would appreciate if you help me keep an eye open on the chat box. Okay. Okay. So since Dr. Grace has said she might be stepping out of away from her laptop for a while, who is the class rep? Ladi, right?
Yes, ma.
Ladi,
Yeah.
will you help me
I'm
keep
doing-
an eye on the chat box? Okay, great.
Yes, I'll do that.
So, for the benefit of those who joined after Dr. Grace has done the introduction, my name is Dr. Chinwe. I'm a senior lecturer in veterinary pathology at the University of Nigeria, Nsukka. I also double as the deputy director of the Vaccine Research Center, where I have my own lab, having just where we have just finished installation of our equipment, and we started our first lab work this week. So aren't you lucky? So it's been a busy week for me. And as she said, I have just returned from the NIH in the US, which is where I got a load of equipment, a container load of equipment, all manner of molecular biology equipment that you can think of, that we have here where I am seated right now in Enugu. It's a center of the University of Nigeria. We are starting off primarily on molecular research and all infectious diseases, and especially the zoonotic diseases. So I've been asked to talk to you about separation of DNA molecules, labeling and detection of DNA fragments. And feel free at any time, general house rules, feel free at any time to interrupt me if you have a question. You can type it, you can say it. I would prefer you say it. Just say, excuse me, ma, and then we will listen to you. If you don't understand anything, ask immediately. Don't keep it to the end. And just to say that I like to have an interactive class. So if you can, if you're in a quiet enough place, you can leave your audio on. If you're not in a quiet enough place, you can mute yourself and then unmute when you want to talk. So I want to have, I don't do lectures. Let's get that one straight. I don't do lectures. I teach. Okay. Does that work with you guys? Are they still there?
That's
Yeah,
fine,
that's
ma.
fine, ma.
Are they
Yes,
still there?
we are, ma.
Eh. Because
Yes.
I want to ejo, how do they say please in Hausa language now? Biko ejo, dala. I want responses, okay? I'm not seeing you, so I want responses. So that's why I said if you can, if you're in a quiet enough place, just leave your audio on so that you can respond when I ask. So I said I don't do lectures, I teach. Does that work with you guys?
Yes, ma.
So
Yes,
we're just
madam.
going to go, we're just going to go on a journey together, okay?
Okay, that's fine.
Right. So in this course, we're going to talk about the fundamental principles and the techniques used in the separation and labeling and detection of DNA molecules. I am sure you've had a class on, wait, just so I keep it in context and I don't take you beyond what you know. Have you had a class on the study of macromolecules essential for life and gene organization? Have you had that class? Can somebody respond, please?
Not really. What we had was more of like a foundation class on the basic structure of the DNA.
Okay.
So the linkages and just some basic tips. So we are yet to
So you
do.
had a class on gene organization in prokaryotes and eukaryotes?
Yes. Yes.
Yes, ma'am.
Structure and function of gene.
Yes. Different. Yeah.
Yes,
Different
ma'am.
types of genes and what they. Yes, we've done that.
Okay. That I need to know that so that I know what I should be telling you. So we're going to talk about, you have learned the structure and function of genes. So I suppose now that you know what a gene is, right?
Yes.
Am I right? Okay.
Yes. Yes.
You know what DNA is.
Yes.
You know what
Yes,
nucleic
ma'am.
acid is.
Yes, ma'am.
Okay, that's beautiful. I have this very lovely, can you still see me?
Yes.
Yes, ma'am,
Okay.
I can.
Can you see what I'm holding up?
Yes.
Yes.
Okay. That's a fine structure of DNA, right? So it looks like this, like sterile, like this. You can all see it, right?
Yes, ma'am.
Yes, ma, we can see that, ma.
Right? Can you?
Yes, ma'am, we can.
Yes, ma'am, we can.
That's the presentation of a DNA molecule that I am holding. I'll be talking a lot of DNA molecules today. Okay. So since you've had that initial lecture, did they tell you what is nucleic acids? Hmm? Did they tell you what is nucleic acids?
Yes, ma'am.
I'm waiting. Can
Yes,
someone
ma'am.
answer?
Yeah. Yes, they did.
I don't know if you can, because I can't hear anybody. Nucleic acids. I don't understand the yes, ma.
Yes, they did, ma'am.
Okay. So can you tell me what is nucleic acid? Anybody? If you put down answer, I will start calling names. So
Okay, let me try.
Tell me what is nucleic acid. Sorry, Ladi, before you go on.
Yes, ma'am.
Do I get it correctly that we have masters and MSc and PhD students in this class, right?
Yes, ma'am.
Okay, I just want to be sure. So go ahead.
So nucleic acid are basically the molecules that makes up the protein structure of the DNA. They are the basic molecules that are involved in the storage and expression of genetic information.
Okay, so you got the second part right, but the first part was wrong. So there are essentially three biological molecules that we call macromolecules. And that includes DNA, RNA, and protein. Okay? So the RNA and protein are generally called nucleic acids. So when we say nucleic acid, it can be RNA, it can be protein. Do you know the central dogma of molecular biology? I'm waiting.
Yes,
Are
ma'am.
you still with me? Okay. Can someone tell us about the central dogma of molecular biology?
Generally, that genetic information usually flows in one direction from DNA to RNA and then to proteins.
That's the central dogma, but there are exceptions, right? We know that there are exceptions. Okay. So we are going to be concentrating on the nucleic acids, which is RNA and DNA. And most of the times, the techniques that are involved in DNA handling is also similar to RNA. But there are a few modifications because they are different chemical entities. Okay, there are differences in their chemistry that lead to a few modifications. So when we say DNA extraction, it could be DNA, it could be RNA, just a few modifications here and there. All right. So I want us to get that clear. So we're going to talk about the theory and the practical applications of the various methods for extraction, electrophoresis, amplification, and labeling. Okay. So at the end of this class, I expect that you will be able to extract nucleic acids from biological samples. Ordinarily, if we were together, we would be able to run a simple lab, but maybe today, because we're doing virtual class, we will also do a virtual lab so that we can see what these things look like. And then at the end of the class, you should also be able to understand and explain the principles that is behind DNA separation techniques and its applications, right? You should also be able to perform gel electrophoresis for DNA separation. We will also do a virtual lab for that. And you should be able to explain the methods of DNA labeling and their applications as well. I expect also that you should be able to design, or at least, maybe not conduct, but design DNA blotting and hybridization experiments, or at least understand what it is about, and then be able to analyze your results and interpret the results. Okay, so let's get to the first part. So now you have received your biological sample, be it, I'm sure most of you already have projects running. Do you? Does anybody have a project running here?
Yes, ma'am.
can you give me an example of the project you're working on so that I can use it to illustrate what I'm talking about? Anybody? Tell us what your project is.
Okay, one of the objectives from my project is to do a molecular work from samples, positive samples taken during participatory epidemiology studies, survey on PPR.
Okay.
So we did a participatory epidemiology, and during the course of the studies, we sampled some animals that were showing clinical signs. So part of it is to be able to identify the type of PPR virus, whether there's PPR virus first, and then identify if which of the viruses is actually in the field.
So what samples did you collect?
Nasal swabs, nasal
Nasal.
and ocular swab.
And ocular swabs. Okay. So you have swab samples. So any other person that has a project running, what samples did you collect? Or are you going to collect? Anybody else? Okay. So we will take an example of your swabs. So now that you have your samples, let's assume you have your samples, your swab samples, the blood samples that you have collected. What do you do with it? The first thing you need to do, if you're going to run molecular techniques on it, would be to extract DNA, right? As we have said, DNA is in every living thing, which is where there is no distinction when it comes to molecular studies across all life sciences, whether it is animal, it is human, or plant. All living things are made up of DNA and RNA and proteins. Okay, so when you come to the molecular level, it doesn't matter whether you are a human or animal or plant researcher. All of this has DNA, RNA, and protein. Okay, so now that you have your biological sample that you have collected, the first thing you need to do with it is to, if you want to do molecular studies, will be to get out the DNA or the RNA, or let's say generally to extract the nucleic acid. Don't forget that sample is made up of nucleic acids, which are RNA, DNA, and protein in all manner of combinations. So, Jonah, can you please mute yourself, Jonah? So, Jonah, could you please mute yourself? Right, good. So now that we have, so you need to first get out your DNA from that. Now, there are different, when you have extracted your DNA, because there is all manner of DNA or nucleic acid in that your sample, and especially when you are working with, okay, let's take, for instance, this nasal swab sample that you have collected. It will have the DNA of the host organism, which is maybe if it's sheep or goat or cattle or dog or human. That swab contains the DNA, the genomic DNA of that, and then the DNA of all infectious agents, be it bacteria, viruses, or parasites that are contained, that organism is hosting at the moment. All right. So you want to be able to get out the DNA, and when you are getting it out, you don't know which one is the host DNA and which one is the infectious agent DNA. Is all DNA that is mixed up in there. So let's just take a quick look at a small demo on how to extract nucleic acids. Can you see this page?
Yes, ma'am.
What are you seeing?
How to extract DNA from anything living.
Great. So, so this is just a virtual lab. There is a gene extraction. Yeah, this is what I was looking for. You can see it, right?
Yes, ma'am.
Yes, ma'am.
Okay, so let's get into the lab. So let's say you have this, they're just talking about all the different applications of, this procedure. You can use it in forensics, analysis of forensic evidence. You can use it in so many other things. It's typically the first step in a longer laboratory process. And it is important because you need to be able to get out, if you're looking for, depending on the downstream application that you're looking for. But in your cases, you are mostly going to be looking for infectious agents, right?
Yeah.
So this is a cell inside which there is a chromosome, inside which there are genes. You can see the long strands of DNA, right? And then this DNA all make the protein. So how do we start? You have collected, don't forget that you can't see this DNA you can't even see a cell with your naked eye. But we need to, this DNA is enclosed in the chromosome, which is enclosed within the nucleus, enclosed by the nuclear membrane inside the cell that is covered by the cell membrane, and then with all the connecting tissues, that hold the cells, different cells together to make a tissue, and then the organ. And this is, you have collected a tissue sample. So we first need to get out that DNA that is inside. So if you take a swab of the oral cavity, for instance, you're scraping off some of these cells that are there, the host cells, which is what I've explained to you before. And now you want to get started with the DNA extraction steps. And so we are assuming we're in this lab right now. And you have all of these things. You have a centrifuge, you have water bath and pipettes, and you have your swab. Now, you take that swab that you have taken, and then okay. It's asking us to. That's the process of getting the swab. And Ladi, you said you already have the swab. Now, in this swab, you have millions of cells. which include the cheek cells, that's the cells of the host. And then you put that in a tube. And there you take your lysis solution. That's the first thing that you take, because you want to lyse the cells so that you will be able to, don't forget all the layers of protection that are guarding the DNA molecules and the RNA molecules inside the nucleus. No, they're first inside the chromosome, which is inside the nucleus, covered by the nuclear membrane, inside the cell, which is covered by a cell membrane. And in the case of plants, it's also covered by a cell wall, right? So if you want to break down all those coverings so that the DNA will be released into the solution, you take your lysis solution and then you add that to your sample, whether it is blood or the swab that you have taken or a tissue that you have macerated, and then you place it, depending on, in this, I'm just teaching you the very basic steps. Usually, practically, you would have a DNA extraction kit. That's what most people use now. But I want you to understand the principle behind those things. So in the kit, you have all the reagents arranged for you. They will say, add 5 mL of this one to this one, incubate for 5 minutes, put this one in there. So they have written a protocol which will optimize your DNA extraction. But for the purpose of this class, we want you to understand what you're doing with that extraction procedure, okay? So we place the tube in our warm water bath and then allow it to lyse all of those cells. See how the cells are, all those protective layers are being broken down, right? You can see it, right? Do I still have people here?
Yes, ma'am.
I want to be sure you're seeing what I'm showing you. Some of these proteins are the proteins that wrap around the histones. Those are part of the proteins that wrap around the DNA in the chromosome. And then the lysis solution just breaks everything down. So with the lysis, you've broken down the cell walls and all the membranes, but you still have the DNA wrapped around the histones. So what do we do to get the DNA out, to get the histones out of the DNA so we can free the DNA completely? Now we take that back to our bench. And then we take a concentrated salt solution. So, oh yeah, we have to add this to the tube. Take the salt solution, add it to the tube, allow it to stay for some time, and then that salt solution breaks down the histones. So now you have a protein there, and then you have the DNA strands inside. Take that into a centrifuge. Don't forget, you will always need to balance your centrifuge. Close it, spin it, and then you have all the cellular debris come down to the bottom of the tube like this. So here you have all the proteins and all the cellular debris, and then you have the DNA in the liquid. We call that the supernatant. I'm sure you know that. So now we take that and then we get out the supernatant and then we place it in a different tube and we can throw away the debris. because we don't need that. Take that supernatant, and then we take alcohol, which precipitates the DNA, and we add that to the tube. Mix that, and mix that, and mix that, and then you see the DNA. It begins to clump together. It begins to precipitate. And now you have the DNA all clumped up in the, all precipitated out of solution. Okay. And then we take that back to the centrifuge. Put it back in the centrifuge. Don't forget to balance your centrifuge. Close the lid. Spin it again. And this time around, because you precipitated the DNA, you can now take out the supernatant now. You have your DNA left at the bottom of the tube. So, does that make sense?
Yes, ma'am.
Great. So this is just the principle of what you're going to do. But like I said, practically, you will always, or most times, not always, because there are times that you would want to try your hands on doing it yourself, depending on what you're doing. But most of the times, because there are companies that have taken time to do these things, optimize them, make all the reagents available, and then they package it in a kit that is called the DNA extraction kit, and they sell to you because they spent their time developing it. You pay more. But if you don't have a lot of money, you can do it yourself using just a lysis buffer and alcohol, and you will be fine. Okay. All right, let's get back to our. So now you can do DNA extraction, right? Yes, Ladi, go ahead.
So I want to find out, the alcohol. Is it a graded alcohol? What type of alcohol do we use?
So it's usually isopropyl, isopropanol or isopropyl alcohol. You would find details of the protocols. You can find details of the protocols online if you want to try your hands on extracting it yourself. But also you can, is this that one? So there's this very fun exercise here, which is really very basic, trying to extract the DNA from anything. You can use peas, you can use beans, you can use anything that is from living thing, blend it, which is trying to crush all those things, and then add a detergent, which will break down which will lyse the cells, and then add an enzyme, which you can use pineapple juice actually, because some they just use meat tenderizer. But there's the enzyme in the meat tenderizer is gotten from pineapple or from pawpaw. There are two kinds of it. And then you put alcohol. So essentially it is the alcohol that separates the because DNA is insoluble in alcohol. So once you add alcohol, it precipitates the DNA. And that is usually 70 to 90% isopropyl alcohol. Okay.
Okay. Then the lysis
who else
buffer.
had their hand?
The lysis buffer.
The lysis buffer is usually made of detergents. That's why. Go ahead. Go ahead.
Okay.
ahead with your question. Go ahead, Ladi. I'm listening.
Okay, no, just wanted to find out the issue of the components of the lysis buffer and the isochron. But most importantly, you mentioned the fact that most of these things come in kits now.
In a
kit, so that makes it more easier and somehow.
I am just telling you the components of the kit.
Yes, so
just in
what
case
I want
you
to
want
find
to
out is
to try.
Why? What is there any limitation of you doing it yourself than using the kits? Is there any, based on your experience, is there any reason why?
Yes, there is.
Okay.
Yeah, why people will want to use the kit? Because the kit has been optimized.
Okay.
When we say optimized in research, it means that they have found the best conditions and quantities to use to get the best result. Okay. If you do it yourself, don't forget, like this experiment now, you are using a lot of peas. You will be collecting samples that you have. If it's bacteria, you have a load of bacteria. If it's parasites, like I work with trips. I work with trypanosomes. In humans, trypanosomes, you might have the parasitemia is so low that you can't even see it. So if you collect 2 ml of blood samples, you might have about 10 parasites in there, right? Or even sometimes less than 10.
Yes.
Okay. And if you extract DNA, you want to be able to get the DNA of that 10 parasites. So going to use anything will not work because, and don't forget, in comparison with the host DNA, you have a lot of the human DNA in the blood. And then you are looking for 10 parasites. So people have taken time to, optimize these things so that you can get out that thing that you're looking for. I think it's just reinventing the way if you want to go back to do it yourself. Although there are circumstances when you want to do it yourself. Like currently, I am trying to see if using a kit and doing the ethanol chloroform extraction myself, which one works better for trypanosomes, because I am having a huge problem with parasitemia. Do you understand?
Yes.
So that's going beyond what you would normally do. But for your level, you normally would use a kit and they should work fine.
Why I'm asking this is, some of these kits now are gradually becoming more expensive. So if the cost implication, going back to the conventional method, will still produce the same results, why not rather than the kit? So
if you quickly Google phenol chloroform DNA extraction, you will get a protocol with which you can do DNA extraction. Very convenient. I mean, very cheaply, but it is inconvenient. Number one, phenol is a chemical you don't want to be handling if you're not, if you don't have to handle it. It's not good for pregnant women. In fact, a pregnant woman should not handle it. And then other things. But if you're strong, I have done phenol chloroform extraction myself because I was looking for RNA and I needed something more stable. Now, part of what you do when you add the this step where we add the meat tenderizer is just a step where you destroy the proteins, the rest of the proteins that are in that solution. Don't forget, biological molecules are DNA, RNA, and protein. And you want to extract DNA or RNA, not protein. So this step of adding the meat tenderizer or the enzymes, which is usually in part of your kit, is to break that. It contains proteases to break down the proteins so that you have only DNA and RNA left. So this process that I've described to you, we generally isolate and extract what is it called now? Nucleic acids. So in that your extraction, you would usually see it more clearly if you're working with bacteria. If you do that extraction and you run it, you will be able to see the RNA and the DNA.
Okay.
Okay. Usually the bacterial ribosomal RNA is so abundant that you can see it always.
Okay.
So, but if you want a pure DNA sample, you want pure DNA extraction, you don't want contamination with RNA, then you add what we call RNases. RNases will break down any RNA in the sample. And if you're looking for pure RNA samples, then you add DNases, which will break down DNA, any DNA in the sample. But if you don't really care whether it has RNA, both, then you can omit that step.
Thank you, ma'am.
Great. Somebody else was raising hand. Saba, was that you? Matthew.
Yes, ma. Yes, ma.
Has your question?
No, not really. I wanted to ask, in the DNA extraction process, the first step, which is the addition of the lysis solution, is this solution selective to its in its activities? Because some cell walls, most cell walls are made up of protein materials, but you have it that they break down, but they don't break down as far as the histone proteins, which later on you will have to add the other step procedure, that's the tenderness for it to break down the histoprotein. I just want to find out, is it that they are selective in activity?
No. You see, the cell walls and the cell membrane. So you have, you know, proteins are made up of, what do you call them now? You know, proteins have a basic unit and a building block, right?
Yes.
So there are enzymes are proteins as well. You know that. But they are small proteins. Now, the cell wall and the cell membrane are very highly organized proteins. You don't forget the cell wall is, they say is oligopolysaccharide. No, how do they call it now?
Yes, that's correct.
So it has both fats, proteins, very highly organized. So what the detergent does, detergent is mostly to remove the lipids. So when it removes those lipid part of the cell walls, it just destroys the structure.
Okay.
All right. So it's like when you have a building, when you are building a house, you get blocks, right? And then you use cement to join the blocks together, okay?
Yes,
right?
Yes, ma'am.
So now when you want to destroy the house, you can either find something that destroys the blocks or destroys the cement that joins the blocks together. So let's say there is a liquid that you can pour on top of that structure and it will dissolve all the cement. When it dissolves all the cement, what do you have? Blocks with holes, right?
Yes, ma'am.
And then anything, if there are rats or lizards inside that house, they can come out from that holes. So that's just what this first step does, to break down the structured proteins that are covering where the DNA is hiding, so that the DNA can come out.
Okay, ma'am. Okay, ma'am.
So it's that oligopolypeptide structure that it destroys. It doesn't break down the proteins.
Okay, ma'am.
Right. Any other questions? All right, let's get back to. So now that you have your DNA extracted, Dr. Grace, I don't know if you're here. This class will probably not be done in 2 hours. We have not even finished slide number 1. Okay. That means we'll continue another time, but let's do what we can do. So now that you have that DNA extracted, where did we find that? So we have this tube with our DNA here. Now, this DNA that is here includes all manner of DNA from all the organisms that were in your sample. Okay? So, but you are looking for something very specific. How do you now know which one is the host DNA or which one is the infectious agent DNA that you are looking for? How will you know? The first thing you do will be to separate this DNA that you have so that you can look at it according to sizes. It is the size of it that gives you an idea of what DNA it is. This is just an idea. You're not sure yet. But it's like having a room full of people or a room full of, how do I describe it now? Okay, most of you are vets. You have a poultry pen. You come into the poultry. There are cockerels, there are broilers, there are these. So the first thing you need to do is to sort them. Okay, let's get a more appropriate example. When you have day-old chicks, right? It's all day-old chicks. But you need to sort them according to sex. And say these ones are this, these ones are that, these ones are that. So the process of DNA electrophoresis is just, gel electrophoresis is just a way of sorting the DNA that is mixed up in this your extract, sorting them out according to their sizes so that you can begin to have a sense of which one is what, okay? So that process is called gel electrophoresis. Now, there are actually two types of electrophoresis before we go on. That's DNA separation. You want to separate the DNA according to sizes. You can do gel electrophoresis. You can do capillary electrophoresis. Anas Zaki, can you please mute yourself? Right. So, but capillary electrophoresis is not a technique you will often use. So I'm going to skip it, and we will talk about, so the DNA separation method that you will always use would be gel electrophoresis. And there are two types of gel electrophoresis. I've explained the principle, which is to sort out the DNA. You have all manner of, several DNA mixed up in your extract. So you want to sort it out according to sizes so that you begin to get an idea of what is what, okay? And it works by passing the DNA through, because of their charge, they have an electric charge. If you look at the atomic and the chemistry and all those things, the DNA molecules are charged. I think they are negatively charged. I'm not sure. I think so. And so passing them through a high voltage, when you pass them through a high voltage in a kind of a sieve, it will start separating according to sizes. Now, the principle of gel electrophoresis is we use a gel matrix, which can be agarose gel or polyacrylamide gel. Agarose is gotten from, I think, a seaweed or something. Polyacrylamide, on the other hand, is a stronger chemical, which you don't want to use if you don't have to use it, because it has its own dangers. But generally speaking, in the lab, you will most of the times do agarose gel electrophoresis. Polyacrylamide you can do usually if you're working with proteins, if you're looking for proteins, and then if you're looking for like very tiny DNA, like very small DNA molecules or RNA molecules, you will use page, because this is polyacrylamide, like have smaller, it's like when you they're just sieves, so to say, they're just sieves. So agarose is a bigger sieve. You can make it up whatever percentage to increase either the size of the sieve. But polyacrylamide is usually a smaller sieve, so it will let only very small particles, DNA fragments pass through. Okay, so that's just the difference between agarose and polyacrylamide gel. We will also get back to our lab and look at how to prepare agarose gel. That's what you will mostly use. Any question on that, or we go to the lab? Okay, let's go to the lab. First close this. We've done this, we've done this. Let's see this. Okay. Let's get back to the lab. Gel electrophoresis. So now we have our DNA that is extracted. Usually, you know what I showed, what you saw looked like there is something in it. The truth is that usually what you will extract will just look as clear as water. You wouldn't know if there is anything inside it, but you're just told or you believe that it contains DNA, okay? So don't be looking for something that is that cloudy as I showed you. That is only if you have used a lot of beans or a lot of plants or a lot of whatever you have. But if you're collecting samples for research, what you will come out with at the end of your DNA extraction will be a very clear liquid that you cannot see anything in. But you think and you hope that it contains DNA. So let's get into the lab. It has a mix of different sizes and portions of DNA. We call them DNA strands or DNA fragments. Now you want to sort them out according to their sizes, as I've explained. They're all mixed up inside this tube. Now, how do you sort them out? It is using gel electrophoresis. Okay, so we get our DNA extracts. So this is what a gel looks like. You can see when you magnify it, it's just a sieve, okay? It just has holes through which the DNA fragments will pass. And I'm going to show you, we're going to see how to, oh wow, they didn't even show how to make a gel. Let me see if I find how to make the gel. No. Okay, so this assumes that you have made your gel. There is what we call a gel casting tray. So the first thing you do would be to heat up your, we normally use PBS to do it, as buffers. There are several buffers that you use. Then you get the agarose powder and then you heat it up. You heat it up and then you pour it. And then it has something like combs, or not like, it's called combs, but it also looks like combs that you use to comb your hair. And then you put it and you pour the molten agarose inside there and then allow it to solidify, to cool and solidify. And when that has done, you now have this gel with these holes that is formed by the combs. Tomorrow. Okay. All right. So now that you have done that, we also use what we call a loading dye, because you don't want your DNA sample is looking like it's water. So you're going to put this gel inside the gel tank and pour in the appropriate buffers. People have researched these things. They have the buffers that you should use. Usually is either tris acetate, TAE, or no, TEA or TBE. So those buffers create the right conditions for the DNA molecules to migrate under high voltage. So you put a loading dye so that your DNA, the extract is just looking like water, and you have the buffer covering this inside the tank. So when you pour it, you don't want it to splash everywhere. So you use a loading dye, mix it with your sample, and then put it in the wells and apply voltage. So once you apply the voltage, the DNA starts migrating from the negative charge to the positive charge. And then look at that. Look at a beautiful picture of how it's moving. All right. So now you have separated the DNA in that your extract into the lengths, the various sizes that they have. Usually when you do this, you have what we call a DNA ladder that you put in one of the wells. A DNA ladder is a sample that is made up of known DNA sizes, so that you'll be able to see, you now have all the sizes in your ladder, and then you can compare that to the sizes of your sample. Now, when you have done that, because you can't see the DNA with your eyes, you have to find a way to see it. And that way to see it is by staining. When you now stain, there are dyes or things you can do to make the DNA visible to the eye. I'm going to talk to you in a bit about that. And then when you've done that, you will now be able to see the DNA that is separated into various sizes. So in this agarose gel now, this is the DNA ladder of known sizes. And these are your samples, which have been separated into these different sizes. Okay? So now we know the size of this, and we can say, oh, our DNA, this one is somewhere about this size, and we can estimate their sizes. So what we do to make it, I talked about the staining. I'm going to tell you a little more about staining now. Oh, wow. I see. So they gave an overview of the process, and now they are giving the details. So first of all, we make a gel. This is how you make a gel. Oh, this is more detailed than the box. So I'm going to leave you to watch. Oh God, what do you prefer? That we do the theory and then you can watch this later, or you want us to get through it together? Tell me what you prefer. Are people still there?
Yes, ma'am.
Tell me what you prefer.
Well, if we can have the slide later, we can follow it up and watch the details so that you can, because of time factor, we can focus on the major things we need to know. So some of the little details, we can follow it up later if you will share the slide with us.
Do we all agree?
Yes, ma'am. It's fine.
Do we all agree? Yes, it's fine.
Yes, ma'am.
So you can watch. Great. So let's get back to business. Right. Okay. The staining and visualization is usually done with, I actually think let's finish this. We will do it very quickly. So you weigh out your agarose. Oh. Put that in there. Add your buffer, which is usually PBS. Put that in the microwave. Microwave it and it melts the agarose. Take it out. Here is your gel casting tray. You pour it in the gel casting tray. That's the comb I talked about. You add the comb in there, and then you allow it to solidify. Then you remove the comb, and then you take that and put it, pour some more. That's the electrophoresis tank now. You pour your buffers, and then you put your gel in there, and then now you can load your samples. You add the loading buffer into your DNA samples and all that, and then you start loading. So you take the loading buffer, are we supposed to click? Okay. So now you take your loading buffer, add it there into your sample. And then you load this is the gel that you made. See that? Because of the loading buffer, it won't splash everywhere. You will still have a bit of a splash though, depending on how experienced you are. Nice job. So now you have loaded your samples. Then you take your standard, which is the lab that I talked about. Take that, and then you load. Okay. Now you connect the electrophoresis tank. Connect that to the power supply, and then you turn on the power supply. you start seeing this kind of bubbles at the down part of it, right? And that means it's passing the current across. Great. So that's how it migrates. It keeps going and going and going. Now it has finished running, and then you take it out, and then you stain. Now, usually in the past, people used ethidium bromide for staining. That is considered carcinogenic now. So people no longer, in fact, in several labs, in several countries, it has been banned as a staining agent. I know many labs in Nigeria still use ethidium bromide, but there are better dyes now that you use. Some of them you add when you are making your gel, something like gel red, cyber green, cyber safe. Those are considered safer dyes. It's not like they're completely safe because they bind nucleic acid. So anything that binds nucleic acid, you should be careful with. But they are safer than ethidium bromide. So in the past, when people use ethidium bromide, you now take your gel and put it into an ethidium bromide solution and let it sit for some time. But now with better stains, you add the stain when you are making your gel so that as it's migrating, you don't have to do this staining at the end of the day. So do that and let it sit for some time. And then you take out your gel. You have what you call a gel imager. It can be a UV lamp. It can be as simple as a UV lamp like this, or it can be as complex as a big box with that is complex that we take all the images. But the basic component is this UV light, which shines. We put your gel on top of it, and then it shines ultraviolet light, which now makes the gel to fluoresce. You turn on the UV light, and you will begin to see there's something like this. So this is what you will get at your image. This is what is your end product for gel electrophoresis. Now, these are the sizes of the DNA ladder that you have used. And so you can hazard a guess that this particular one in your sample, okay, actually they have that assignment for us. So what would you say is the size of this? Everybody, I want answers. What would you say is the size of this one?
pp.
Considering the sizes of your great 6,000 base pairs, what would you say is the size of this one?
Sometimes around 3,005.
Is it
3,300.
3,000. I got me in a class. Where's the Omoronja no nearby? Omoronja nearby. I'm in a class, okay? I'm in a class right now. So settle with that.
Maybe 3,003 or 3,004.
That is my life now. I will do both academic, and it is an electrician that came to work that is calling me to come and see. You see my life now. I have to do both academic and non-academic and even technical. God will help me. Go on. What size?
3,003.
Good. It's about 3,003, 3,400. It's not up to 3,500 because it's not really halfway between this. But that's acceptable. Even if you say 3,500, that's acceptable. What would be the size of this one?
1,004.
Good. Or 1,005. So now the sizes are measured in base pairs. In base pairs. And if you remember this that I showed you, right? You can see that. This is one base pair. This is two base pair. Those lines are crossed like this. They are considered base pairs. And that is how we know the length of a DNA fragment. So when you say it's 1,000 base pairs, it means it has like this into 1,000 places. Okay, and that just tells you how long the fragment DNA is. All right. Oh, we have to write it to be able to move on. So say 6,000. What did we say here? 3,000.
3,004.
And here
1,005, 1,004.
1,500. Okay. Let's go. Wow, it says all of our estimates are correct. 6,035,15. You see? We got it. You are all geniuses. So now we are done with gel electrophoresis. I think we are done with that. We've done all this, right? Yep. We are done with this. Okay, so let's get back to, we've done this, we've done that, we move. So labeling of DNA molecules. Now, when you want to find certain DNA molecule, like that sample that you have taken that we've extracted and we said it contains both the host DNA, it contains the infectious agent that you're looking for, it contains the infectious agent that you are not looking for. And we have separated it, and we have separated it according to sizes. Now, let's say you are looking for a bacteria that you know that the DNA size that is, the DNA size that you're looking for is, say, 600 base pairs. But there are many 600 base pairs in that, your sample that you have separated. How do you know which one belongs to what you are looking for? Say you are looking for Pseudomonas. How do you know that this one is the one for Pseudomonas and not for E. coli? That is where DNA labeling comes in. And there are several ways you can label DNA. You can do radioactive labeling, and you can do non-radioactive labeling, which is you can do by fluorescence or biotin, or I don't know what this DIG is. So in the past, it used to be radioactive labeling, but most people are moving away from radioactive labeling now. In fact, to do radioactive labeling, in certain places, you have to have extra authorizations. Like for me, when I was doing my PhD in the UK, my supervisor didn't have the authorization for me to run radioactive labeling in our lab. And so I had to go to Sweden to go and run the radioactive labeling and detection techniques that I was running. So most people are moving away from that because of the health implications. So most of the times now we use fluorescent labeling, and there are several ways you can label DNA. It's usually not something you need to do by yourself, except you are using it you are applying it in a technique. So sometimes you can get some, there's some random priming, end labeling, PCR-based labeling. There are companies that can do this for you. If you're looking for a particular, we call them probes now. So you want to find a particular type sequence. All right. I think I need to explain what sequence is. So DNA, how do I start with this now? So those base pairs that I showed you, right? There is one on this side, there is one on the other side, and they pair. Now, those bases, there is a base here, there is a base here, and there's these two pair. And that's where you have the ACGT, okay? And A can only pair with this, C can only pair with this, G can only pair with this. So that ACGT is what we call a sequence. That's the sequence of the bases in the DNA molecule, right? So if you're looking for a particular sequence, and we have to know that every organism has, every DNA has its own unique sequence. So if you're looking for an organism, you have to know the sequence of the bases in the organism that you're looking for. And then you can make a probe that you now label, and they use it in an application so that it can find similar sequences in your sample. That's the principle of labeling DNA. Now, there are companies that make probes. They can do end labeling where they tag a fluorescent molecule to the end of the DNA sequence that you have given them, or there are other means of labeling. That I will not dwell on because it's not something that you will usually do. But what you will usually do is blotting techniques. And that is just what I've described. When you are looking for a particular sequence, you have this DNA that is mixed up with DNA from the host, from various organisms that you don't know. But in that mix, you want to find the DNA of Pseudomonas. So what you will do is to make a probe, get the people that manufacture DNA or primers or whatever to make you a primer, a probe. A probe is just an oligonucleotide sequence that you know is complementary to a section of the Pseudomonas DNA that you are looking for. So the Pseudomonas DNA is like this. You make a probe that is complementary to it, that can bind to it, right? And you have it labeled with some form of detection, labeling technique, probably a fluorescent label added to the end of that molecule. Now, that is the principle of DNA blotting, because you're going to use that probe. It's like looking for a needle in a haystack. How will you find a needle in a haystack? It's simple. You get a magnet. Once you get a magnet and move it around that haystack, it will magnet the needle, right? That's what a probe does. And that's what you do in DNA blotting techniques. So you're going to find a particular sequence of DNA in a mix of several DNA. And then for DNA in particular, we call that southern blotting. But for RNA, we call it northern blotting. There is also a similar procedure for proteins, which is called western blotting. These are just, I think northern blotting was gotten from the name of the person that, or no, is western blotting or both? I don't know. That was taken from, is southern blotting. I can't remember which one. It was taken from the name of the person that started using the procedure. And then the other ones were just, randomly chosen. So southern blotting for DNA, northern blotting for RNA, western blotting for proteins. All you need to do is to prepare your gel and transfer the DNA to a membrane, which is like your agarose. No, not agarose. There's what we call membrane, is nylon membranes. So you have your sample, you run it on a gel, as we have said, electrophoresis, separate it into sizes, and then you take that gel, transfer it onto a membrane. I'm not sure I have I don't think I have a lab for a virtual lab for this. I would have loved to show you. I did northern blotting for several months when I was doing my PhD. It is a laborious process, although there are things to make it easier now. but it is a perfect, like if your experiment works with northern blotting, it is, nobody can dispute it, right? These days, a lot of people don't do it because it is laborious. They will rather use qPCR to do it. qPCR is more sensitive, but it is not very specific. But northern blotting is very specific. If you find it, it is there. If you didn't find it, it is not, well, if you find it, you are sure. You cannot, nobody can question whether it is there. So it is very specific. So you prepare that, transfer it onto a nylon membrane, and then you put your probe in a solution, put that membrane. That probe will just go around the whole membrane, which is your DNA that you have separated, and find which DNA is complementary to it. It will bind to it. And then you can now develop it with the fluorescent developer and then go and see it. Now, when you look under, you are going to see where your probe is. It means that that probe that you designed for Pseudomonas, once you have it emitting signal there, it means that Pseudomonas is there. I hope that explanation makes sense. If you have a question, please feel free to ask. We can actually finish the class, I think, today. Are there people still here?
Yes, ma'am.
Yes, Matthew, go ahead.
Okay, ma, thank you very much. My question is actually on the fact that DNAs are actually paired molecules, and in this sense of doing the DNA blotting, I was wondering how possible is it to get a complementary, that's using the probe with bases that complement with that of the DNA you're looking for to pair. Because I was expecting that the DNA, maybe there could be a process where we separate the DNA molecule because they're actually wind up and paired and wind up. So maybe we'll separate, then would be able to, I don't know if.
So before you start blotting, you would have separated on by electrophoresis.
Okay.
So one of the things electrophoresis does is DNA is a spiral molecule, but when you run it through a gel, it straightens it up. It makes it linear. Okay?
Okay.
Okay. So you must have separated on a gel. It is that gel, it is from the gel that you transfer to the membrane. And so it's on the membrane according to the sizes. Okay? And then the probe, you design the probe yourself, knowing what you are looking for. So usually, you know you are looking for a certain sequence. There are applications that before they used to design it by eye and, manually, but now there are several applications. Sorry, I need water. Now there are several applications that you can just use to design a probe of primers. But what those applications do is that you have to supply the sequence of the DNA you are looking for. Okay, so if you are looking for, say, E. coli, you are suspecting colibacillosis in that sample. Or, Ladi, what is that thing that you're looking for in your sample? PPR. You go to gene, there are several databases like the NCBI. You find, and there are diagnostic markers or gene sequences that are peculiar to certain infectious organisms that we use to find them. So you go take a sequence of a portion of that gene of that particular organism that you're looking for, put it in that program. It will design an oligonucleotide that will be complementary to it. When we say complementary, it means it can bind to it perfectly. Okay?
Okay.
And now that's your probe. And that probe is labeled with a fluorescent tag. It's like it's tagged with a fluorescent molecule at the end. So when you do your hybridization, which is now putting that probe in solution to hover around the gel, the membrane that you have transferred your DNA to, when the probe is circulating, it will just find the sequence that is complementary to it and bind to it. And so once it's bound to it, there are several other steps to make sure that your DNA is trapped to the membrane, and then make sure that the DNA is also straightened up and is ready to accept a complementary sequence. So once your probe comes and looks around and it finds a complementary sequence of DNA in that membrane, it will bind to it. And when it is bound, you now take out the membrane. That you do that incubation for some time. All the protocols have been written. You can now modify according to what you need. You take out that membrane, and then you have a developer that will now develop the fluorescence. So when you develop, if that probe was bound, there will be fluorescence and you can see it. But if the probe was not bound, then you won't have fluorescence. It means that that's the section, that particular thing you are looking for, that you have developed that probe for, is not in that sample. Does that make more sense now?
Yes, ma. Thank you very much, ma.
Good. So this is a schematic diagram of southern blotting. Okay, so this is the DNA that you have extracted. You run it through Agarose, you separate it into then you take that and blot it on a membrane. This is the nylon membrane. This is your gel. You put it there. It gets transferred to the membrane. You have the membrane with the DNA, and then you add your probe, and then the probe goes to find the complementary sequence, and then you expose that to in the past, you normally would use X-ray. These days, they use fluorescent tags, and then you find this because the probe has a tag that will. So if it bound, you will see it. If it didn't bind, you will see nothing. So maybe we will copy this image and put it here for you. Okay, I'll do that later. So I suppose we understand that, right? Now, the detection methods, I think I have actually talked about this. After you have done your blotting, the detection method for autoradiography, if you use the radioactive label, right? That's the one I said. Most people now don't do radioactive labeling, but you can still do radioactive labeling if your lab is equipped for that. But most people will use chemiluminescence or fluorescence now. So that's the more detection methods that are used. And so if you have, let's get back to here. So you now have this. How do you interpret this? Sometimes you have this, you have other fainter bands, okay? There are programs that you can use to quantify the band intensity. And that tells you, that gives you a relative measure of the amount of what you're looking for that is there. So that's how you analyze your results and quantify the DNA that is there. So you do quantification by a measure of the band intensity. There are programs that you will use, and then you put your image, and then it will quantify. You will give it a quantity. You will quantify the intensity of the band, And that's directly proportional to the amount of that DNA that you have in that sample. Okay, I think we are actually done. That is impressive. So, I think this is the last slide, but it's going to take us a little bit of time. So, we didn't talk about capillary electrophoresis. I told you, you probably don't need that. Gel electrophoresis is what you will mostly do. Now, other techniques that we can apply to the DNA that we have extracted include real-time PCR, or let's just say PCR. We haven't talked about PCR. That's true. I omitted that. So I have a virtual lab for PCR. Let's get back to the lab. PCR, no DNA extraction, electrophoresis. Yep, this is PCR. You will always be hearing PCR, PCR, PCR. You cannot do most molecular techniques without doing PCR. What is PCR? Anybody knows? It's just an abbreviation. What's the full meaning?
Polymerase chain reaction.
Correct. Polymerase chain reaction. And we are going to see how the chain reaction goes.
The world is awash in DNA. It's in all living things and the things they leave behind, like skin flakes, hair, secretions, and feces. But an individual strand of DNA is so small you can't see it, not even with a microscope. So how do scientists visualize it? One way is with PCR. It's a technique that lets you focus in on one specific bit of DNA code. It's based on the natural process that cells use to copy DNA. That's the P for polymerase. But it adds a twist that lets you make copies by the billions. That's the chain reaction. Since everything living has DNA, PCR has tons of uses, from genetic engineering to forensic identification and medical testing. The only catch is you need to know the sequence of DNA letters around the bit that you want to target. Click or tap to show the DNA in the sample.
So you hear that, you need to know the sequence, and you don't have to know it often. There are websites and databases where you find the sequence of the DNA you are looking for that organism, that infectious agent, that bacteria, that parasite that you are looking for. Okay. That's your target sequence. There are sequences that we already know that it is only E. coli that has this sequence, or it is only Plasmodium that has this sequence. And so when you go to look for that, once you see it, you know it is Plasmodium is there. That's the principle of target sequence. And that's what you will use to design whether it is a probe or it is a primer. So probes we use for blotting, primers we use for PCR.
That's so you can design primers. These short pieces of DNA will attach on either end of the target.
So the only difference, because I know some of you were getting probes and primers. Probes is when you are trying to probe, you know, that's for blotting. A primer is what you use to prime, to say, or you start making more copies of this. Okay. But all of them are means of finding the particular sequence of DNA that you are looking for. In that, your mix of DNA that you extracted, which contains both the host DNA and all kinds of microbial DNA.
One primer binds to each strand through complementary base pairing. Drag primer 2 to its complementary sequence.
So this is primer 2. So we should drag primer 2 to its complementary sequence. Who knows where we will find its complementary sequence? Can you tell me?
It's up to the left, the upper strand to the left.
Upper strand to the left. I think I disagree. Is the lower strand.
Primers are what make
Oh,
PCR
okay.
specific. They are custom made for each target. Different targets need different primers.
So let's get something clear. Unlike probes, primers, you have to make 2 primers. Now, remember the DNA sequence is 2, which is how they now do base pairing, okay? For a probe, part of the process of blotting will straighten the DNA and make sure it's single-stranded. Single-stranded means it's detached from the other strand, so that your probe will come and bind comfortably and it's detached. But in PCR, because you are using a double-stranded DNA, it's double helix. Most DNA is double-stranded. Unlike RNA, RNA is usually single-stranded but gets folded to be double-stranded. Now, because you are using double-strand, the DNA, you are looking for DNA that is double-stranded. You have to make 2 pairs of primers, 2 primers rather, 1 pair of primers, which is 2 primers. You make a forward primer and a reverse, a forward primer and a reverse primer, which means a primer that can find on one strand and the other one that can find on the reverse strand. Okay? So when you make a probe for blotting, you are making 1 probe. But when you make a primer for PCR, you are making 2 primers, a forward primer and a reverse primer.
To do PCR, you need to add many copies of the 2 primers, plus a few more things.
The few more things are just buffers and salts that will create the right condition for the reaction to take place.
DNA polymerase is an enzyme that copies DNA.
So we add the DNA polymerase, which is the enzyme.
Nucleotides are DNA building blocks.
The nucleotides, we call them usually DNTPs. So you add that.
And you need things like water and salts to mimic the conditions inside a cell.
All right.
The rest is biochemistry. And you regulate the process through repeated cycles of temperature changes. To begin cycle 1, raise the temperature to 95 degrees Celsius.
So once you have mixed your reagents in that tube and you put it in a thermal cycler, which is the PCR machine, you set the conditions. The first thing you do, because your DNA is double-stranded like this, the first thing you do is to what do they call it now? To separate it, to separate the strands. And so to begin, we set it at 95.
This temperature is close to boiling. It disrupts complementary base pairing, and the 2 DNA strands come apart. Next, lower the temperature to 50 degrees.
This thing I'm doing now, increasing the temperature and lowering it, is what you set in the program for the thermal cycler, which is the PCR machine. You set that as a program. And then
Complementary DNA strands can rejoin at this temperature. But since the primers are present in much greater numbers, they will lock onto their complementary sequences before the longer strands can come back together. Now, raise the temperature to 72 degrees. This temperature activates DNA polymerase. It attaches at the ends of the paired primers and extends them. It moves along the single-stranded DNA, adding complementary nucleotides.
you see now you started with 1 copy of the DNA. Now you have 2 copies.
It goes until it falls off, reaches the end of the strand, or until the next temperature change. Cycle 2 repeats the same 3 steps. At 95 degrees, the DNA strands separate. At 50 degrees, primers find their complementary sequences. They bind not only to the original target DNA, but also to the products from cycle 1. At 72 degrees, polymerase extends the primers, adding complementary nucleotides as it goes.
Now we have 4 copies.
In cycle 3, the steps repeat again. For the first time, the desired products appear. These are shorter copies of just the target sequence. Click or tap the 2 desired products.
Oh. Shorter copies of just the target sequence. I don't understand what they mean by that. Oh, yeah. Here we have that.
There are just 2 copies now. Watch how the following cycles cause a chain reaction that makes the desired product start increasing exponentially. At the end of cycle 4, there are 8 copies of just the target sequence. And now there are 22. The target copies outnumber the longer ones. We'll fast forward through the rest of the cycles. After 30 cycles, there are over 1 billion copies of just the target sequence and only 60 of the longer copies. The target sequence overwhelmingly outnumbers the other products.
So you see how I told you that the host DNA will be more in your sample than the DNA of the infectious agent that you're looking for. So let's say you have a million copies of the human DNA that you in the blood that you have collected, and you have 4 copies of the plasmodium or the trypanosome that I'm looking for, right? Now, PCR is an amplification technique that we take that 4 copies and amplify it into a million or billions of copies, so that you will see it easier than you will see the host DNA that was there. So at the end of a PCR reaction, you have millions of your target sequence amplified.
Or if the target sequence wasn't present in your sample, you won't have any products.
Exactly.
After all, PCR can only amplify something that's already there.
Exactly. PCR can only amplify something that is there. So if the target sequence is not there, you have nothing. But if it is there, it amplifies it and you'll be able to see it.
Now, you can analyze the PCR sample to get a test result, or use the copies in an experiment, or in any number of ways.
So when you are done with the PCR, you can run gel electrophoresis with your product to see if your target sequence was there. And then you can also use the product for a lot of other downstream applications, such as sequencing. You can now take that and take it to a sequence facility and sequence it. And then you can see the sequence of the bases in that your amplified product. You can also use it in a lot of other applications, but these are the most commonly done ones. But once you finish a PCR, you have to run it on a gel electrophoresis to see whether your target sequence was there. Okay, that's done. Now that we have talked about PCR, there is another technique that is called real-time PCR or qPCR. Q for quantitative. Q for quantitative. Now, real-time PCR is not RT-PCR. RT-PCR is reverse transcriptase PCR, which is what you do when you are starting with RNA samples. So with RNA samples, you have to reverse transcribe it to DNA before you can do PCR. That's RT-PCR. That's for RNA samples. Real-time PCR is a technique that instead of, what I showed you now, what I showed you here is called conventional PCR. So instead of waiting to see the end product at the end of the procedure, real-time PCR gives you a real-time result, like it tells you in real time how much of the DNA you started with has been made and has been amplified. So you use it, that's why it is called qPCR is for quantitative PCR. So what that helps you to do is to quantify the amount of your target sequence that was in your sample. That's another application of advanced application of PCR. And a real-time PCR machine costs as much as $42,000 to $50,000. That is about 75 million naira. So not many places have it, but luckily I have two. So if anybody needs to run qPCR assays, you are welcome to my facility to use it. And then other things you can do with your DNA is like after PCR, you can do next generation sequencing, which is to sequence the samples. You can also do other CRISPR-based methods for DNA detection. CRISPR is the in thing now, which is this enzyme that cuts and joins DNA. So you can cut out what you don't want, edit DNA, put what you want, remove what you don't want, modify what you want to modify. Science is going, molecular biology is going crazy these days. Soon, one day, they will be able to take somebody's gene, modify it, put back, and okay, so that is, I think, all I have for you for today. Do we have any questions? Any closing questions? Or do you have something you expected to learn that I have not talked about? Or you have question about what we have talked about or what we have not talked about? Any closing questions?
Actually, we
Or if the target sequence wasn't present in your sample, you won't have any products. After all, PCR can only amplify something that's already there. Now you can analyze the PCR
Ladi, you were saying
to get a
something.
test result, or use the DNA copies in an experiment, or in any number of ways.
Ladi,
yes, ma'am.
you started saying something but didn't finish it.
Okay, I was just saying that the slide you
got cut off again.
whenever possible for some of us that want to do molecular works.
I didn't hear you.
Can you hear me, ma, now?
Yes, I can hear you now.
Okay, you heard the part where I was appreciating you?
No, I didn't.
Oh, so I said we really appreciate your effort and the knowledge imparted. Thank you so much for what you've been able to do within the shortest within the time possible. We hope that maybe we can organize other class through our lecturer, then we'll share more knowledge with us with regards to molecular, practical molecular studies. But basically, the slide you used containing most of the information you shared with us, we want to have it, especially the first slide that has your email address and your phone number, so that any of us that wants to do any level of molecular work, we can reach out to you for mentorship, for guidance as well. Thank you, ma'am.
You're welcome, and I'm happy to do that. I'll send this slide across and share it with you. But that would not be now. I have a friend, a very good friend of mine, being buried tomorrow. So I am actually just doing this because I love teaching. I'm not in the right frame of mind to do any deep academic work right now. So I will be there at the funeral tomorrow, God willing. And then by next week, I will tidy up the slides and send it to Dr. Grace, and she will send it across to you. Like I said, I have a facility here that have all of the equipment that you will need to do any level of molecular, at least the basic level of molecular work. We can do some degree of genotyping. What we cannot do, we can send to our partner lab at the NIH. They're happy to do that for us. But at least you have basic equipment like the PCR machines, you have the qPCR machines, that's the real-time PCR machines. We have loads of fluorescent microscopes. We have other things that you would need for any level of, basic level of molecular work. And you're welcome to use our facilities if you need it. And I also would be happy to help you design your experiments if you're going to be doing molecular work. I can do that for you. I will basically be available to teach you. I just want to build capacity. I want many of us to know these things so that we can do our own things, because we have lots of infectious diseases that is plaguing our people that we can solve with dedicated research. And the more people we train to do these things, the better for all of us. So I'm happy to train you.
Okay. So you can also help us maybe in addition, put some areas of your research.
Sorry, you got cut off again. You got cut off again. Ladi, we can't hear you anymore, or I can't hear you anymore.
This person is calling me and I'm doing virtual.
Okay, now I can hear you.
Okay, you can hear me now?
Yes.
Yeah, just to say that if you can help us with your area of specialization, so that as many as want to do work, I know you do a lot of work on PPR, and then now you mentioned TRIPS. So let's know most of the areas of your study so that we can key in and
so my
get some level of
my area of specialization is molecular biology. So I have a DVM and I have a PhD in molecular biology and microbial genetics. So I generally can work in the area of molecular studies or genetics of microbial diseases. But I have done work on PPR. That was just a student's work. It's not really a major research work for me. But my own major research work, I do with bacterial genomics. I do with parasite genomics. I work on TRIPS, trypanosomes, human and animal trypanosomiasis. I'm actually investigating currently. My running grant is on investigating genetic exchange between human and animal trypanosomes in Nigeria. So if anybody is willing to, wants to join that, we want to be able to cover as many locations in Nigeria as possible to screen humans and then animals, and then find the parasites, do deep sequencing for the two, and see whether they are exchanging genetic materials. If anybody wants to take that up for any location in the north, I am happy to bring the person on board. That's a running project for me now. I also do work on leishmaniasis. I just recently repurposed an antiseptic for treatment of cutaneous leishmaniasis. We don't have that in the south, but I know there is a lot of it in the north. We're looking for people who help us to collect samples so that we can sequence it and see what species and strains of leishmania that we have in Nigeria. And then we are planning for a clinical trial for the drug that I have proposed. So if anybody has access to hospitals where there are leishmania patients or even communities where there are leishmania patients, we're happy to partner with you to get that work going. Those are the two major projects I have at the moment. But outside of that, we can help you design projects on any infectious disease that you are looking for, that you are interested in. We're also quite interested in Plasmodium at the moment, but that's not, we don't have a running grant on that right now. So the two projects on trypanosomes in humans and animals and leishmaniasis are the ones that are sponsored at the moment, and we have funds that are running at the moment. The other things we can, if you have any other interest in any infectious agent, we can help you design those work. So if you look, if you Google my name, you probably won't see anything on trypanosomes yet, because it's a new area that I started doing 2-3 years ago. I'm still working on my first publications. Okay, you may see something on a preprint that is online. But that's just to say that we are coming. So you might not be able to see, but what you will see is a lot of work on E. coli and bacteria. That's what I have done a lot of work on. But those were my PhD work and other things. But currently, the two projects I have running currently is on trypanosomes. So if anybody is interested in trypanosomiasis or is interested in cutaneous leishmaniasis, shoot me an email and we can bring you on board on the projects. Okay. So do I take it that we are happy to stop here?
Yes, ma'am.
Great. Abel, do you have a question?
Yeah, not a question actually, but a request. I haven't heard about. Not a question per se, but just a request. I haven't heard about the email you made mention of. I don't know if it's possible. How do you, or so that we can get it?
Sure, Dr. Grace will send it across to you, but it's my first name, Chinwe.Chukwudi @unn.edu.nj. I can put it in the chat if you want it now.
All right. Thank you.
Oh dear, I think I sent it to Grace. I want to send it to everyone. There you have my email, you have my phone number in the chat. Any other thing before we close? Are we good?
Excuse me, please, I want to ask a question.
Yes.
Okay, after the second cycles, while making those sequences, raising the temperature, will it unbind the already formed ones, or they will still be intact while making so?
It will, so you go through the cycles, you go through the cycles of why am I forgetting what they are called? Hold on one bit. Cycles of PCR. Denaturation, annealing, and extension. That's what it is called. So you take, you don't do this raising the temperature yourself. You set it in the program. The machine does it by itself. Okay? So you set it in the program, you set the denature, that's high temperature, the 95, it will denature the strands, separate them, and then the system, the machine lowers it again, and then it anneals to the primers, and then it extends, and then it goes back to denaturation. So when it denatures, it will denature everything, both the products that have been formed, and then more. That's why you add a lot of the primers, so that it can bind to as many copies as are being made. Does that clarify it?
Yes, thank you very much.
You're welcome. Dr. Grace, are you here? Doesn't look like she's here. Okay, in the absence of any other question, I would say it was nice meeting you all today. And do let me know if you need any more help. Okay?
Thank you so much, Martha. Thank you. Thank you.
All right. Bye-bye.
Bye. Yeah.